Hello guys. Hello guys. Today our topic is generation and conduction of now impulse. Okay. What is electrical imbalance, electrical charges going to take place in the now. Okay. Yeah. Neurons are excitable, as we know. Neurons are self-excitable. Okay. The membranes are polarized state. The neuron membranes are polarized state. What is polarity? We discussed. Okay. Do you know why membrane neuron is polarized? Why? Okay. We discussed. Okay. Yes. The different type of ions channels we discussed. Different type of channels we discussed. The why neuron is polarized we discussed. The membrane which is self-excitable we discussed. Okay. The ion channels are selectively permeable. Certain different of ions we discussed, sodium and potassium. Okay. Yeah, we all over discussed the things we did here. Yeah, we are going to discuss what is going on, the conduction of now impulse. Conduction of now impulse. Okay. Today our topic is conduction of now impulse. Conduction of now impulse. Conduction of now impulse. Before that, we need to know the polarized state of cell. The polarized state of cell is minus 70 MV. This is resting potential. Resting potential. Potential means certain electrical charge, negative electrical charge. Resting potential is minus 70 is negative electrical charge. Charge inside the cell. Inside the cell. Which cell? Neuron cell. Inside the cell, which is permeable of the membrane, which is sodium and potassium channels are there. Sodium and potassium channels are there. These channels we can call leakage channels. Leakage channels. This leakage channels, when it is leakage in electrical imbalance going to take place, sodium going to out, 3 sodiums are going to out into the interstitial fluid and 2 potassiums are enter into the cytoplasm, mean exoplasm within the cell. In this condition, positive ions are going to out more. That is one of the reason why the 70 MV, why resting potential is minus 70 MV. First case, 3 sodium ions going out from the cell, going out from the cell, 2 potassium ions coming into the cell, 2 potassium ions come into the cell. Ion coming into the cell. This is the one of the reason for why it is maintained minus 70 MV, why it is maintained negativity within the cell. Second reason, as we discussed already, this is quick review further. Second reason the negative ions and proteins are unable to leave the cell. Mean the negative ions and negative proteins, they do not go out from the cell. That is why minus negativity is going to take place within the cell. And third, in the cell there are potassium ions are more and they are permeable in that condition they exit out from the cell. Sodium ions are impermeable into the cell. Sodium ions are impermeable. These three conditions, guys, they describe why it is maintained the resting potential is minus 70 millivolts. This is the reason why it is maintained resting potential minus 70 MV. So, this minus 70 negativity charge going to involve in the now conduction and impulses. Okay? Yeah. How it is going to take place? How many channels involved in this? Okay? There are certain channels which is they can allow the negative ions into cell, positive ions into cell. Mean there is a exchange of negative and positive ions through the channels. What are the channels? Okay? What are the channels? First one is leakage channel. Second one is leakage channel about sodium and potassium pump. Voltage gated channel. Second one is voltage gated channel. Third one is mechanical gated channel. Fourth one is chemically gated channel, mean ligand gated channel. Okay. If come to the leakage gated channel, potassium is permeable through the channel and sodium is impermeable through the channel. Voltage gated channel open when there is any electrical changes going to take place within the cell, any electrical changes. Minus 70 is the resting potential. Okay? Yeah, any impulse, any now impulse mean any electrical charges, any information could go through the now, there is a electrical changes are there, mean minus 70 is the resting potential, turn into the another like number, mean another potential which is above minus 70 or below minus 70. In that condition, if the voltage is changed, that is millivolts of minus 70, if the change, this channel is open. When any changes in voltage charge, voltage, any minus charge or plus charge. Mechanical gated channel open when if any mechanical pressure in the cell applied. Ligand gated channel, any chemical release to activate the channel. These 4 channels are involved in the conduction of now impulses. Okay, we are going to discuss now about it. Okay, I hope, are you clear this? Just look into this. Yeah, mechanism of generation of now impulse, it is conduction along with axon. Mean there is, if there any signals pass from the starting of the axon, there are millions of neurons are there in our body. If any action is going to take place within the peripheral nervous system, any part of body, that is the signals carry forward to the like spinal cord and towards brain. Brain can analyze what happened, brain give the signal into the target organ, target tissue. In that way, there is like signals going to pass through the neurons. How they can generate? The signal is how generate. Okay, this is the topic about throughout the axon. There are billions of neurons, mean billions of axon is there to cross. If an electrical charge, the cable can carry the electrical signals, cables. If come to the human body, the neurons, they can carry the signals, but there are billions of neurons, unlike one single cable in the current. In the electrical circuit, there is a single cable throughout the circuit, they can give the charge end part up to the end part. But if come to the human nervous system, there are billions of cells, neuron cells. They can transfer the signals from one neuron to next neuron. How the signals are impulse, the wavelength of the signals, how they can generate. Okay? Yeah, what is the mechanism in that, we are going to discuss in this topic. Okay? Yes, mechanism of generation of now impulse and conduction along with axon, mean complete axon, how the conduction is going to take place. When stimulus is applied, mean first where there is a start of the signal, okay, signal at the site A, we can take the parts in the axon, site A, site B, site C. Axon is the lang, it is the lang part of the neuron. That part we can divide according to the wavelength of that electrical impulse. Okay? Yeah, first it is polarized toward in the membrane, it is polarized in the membrane. The membrane at the site A become freely permeable to Na first. Okay? These are all over electrical impulses in the membrane, like axon membrane है ना, membrane के अंदर there is a place, membrane place, they can generate the action potential changes, the wavelength throughout the axon membrane there is a wavelength wavelength signals are passed throughout the axon. Okay, we can divide A part, B part and C part throughout the axon. We will going to discuss about it. Okay, first what are the ions are impermeable and what are the ions are permeable throughout the axon membrane, we will discuss. This leads to the rapid influx of first Na is allowed. Okay, the outer surface of the membrane become negatively charged, okay, and inner become positively charged. What happened in this condition? We will going to discuss it. Okay. Yeah, how the conduction of now impulse, how the conduction of now impulse go through along with axon. Okay, this is the question. Okay, guys, any information, this is the axon, the long tube part, and this is the cell body of dendrites, and this is the axon end part. Once signal generated and the signal go through towards the axon, one neuron, it is they can pass the signals, they can pass the electrical charge from one axon to next axon. This is the next axon and this is the next axon. Throughout the body, the signals pass throughout the neuron cells. This is the axon is the long part, which is neuron part. They can pass the signal through the axon membrane layer. They can pass the signals throughout the membrane layer. Okay, how the signals pass throughout the axon and give the signals next axon, next neuron, and they can give the signal into the next associate neuron. This is the pathway which is the conduction of now impulses. They generate here, and they can pass the signals throughout the neurons. There are billions of neurons, they carry information from one neuron to next neuron. Okay, what is going on in the axon, how they can generate the potential, how they can generate different potential. Okay, we will going to discuss in this topic. Okay, yes. Guys, I am going to take axon, long axon. This is the axon, long axon part, this is the cell body part, axon part. Okay, yeah, the resting potential means there is no impulse started, there is no signal started yet. That is resting potential is minus 70 MV, right? Minus 70 MV is resting potential. In this condition, there is no signal pass throughout the axon in the now impulse. Okay, yeah. First, what happened if any action is going to take place? If any mean if any cockroach or any mosquito lays on your hand. Okay, if I beat the mosquito or any cockroach here, there is a pressure is going to created in this muscle. If the pressure is created, there is a signals they can carry through hand to into the brain. How the signals carry from hand to brain? Okay, so certain impulses started here within the hand, some impulses started hand. If we can hit the mosquito, okay, there is channel open first, mean pressure is going to create a, right, that is mechanical pressure is going to created. In that way, mechanical gated channel open. This part, this segment, if any pressure is created here, mechanically pressure, there is a mechanical gated channel open. If the mechanical gated channel open, what happened guys? Just look into this. In this is the cell body as we discussed. How it is minus 70 maintained, mean the interstitial fluid always has sodium more, mean the out of the axon, this part always has more sodium is there as compared with inner. Inside there is potassium is more, that is why it is maintained minus 70 MV as compared with inner fluid, cytoplasm mean exoplasm, there is more Na is outside, mean outside atmosphere, like interstitial fluid mean outside the cell there is more Na is there. If any channel open, that sodium always trying to enter in. If any mechanical gated channel open initially, what happened? This sodium is trying to enter inside. Sodium is trying to enter inside. Sodium is trying to enter inside. The resting potential is minus 70 MV. Resting potential is minus 70 MV, right? Minus 70 MV is resting potential. Resting potential. Okay? Sodium enter. If sodium enter, sodium is enter. If the mechanical gated channel open, sodium is enter in this segment. This is A segment. This is A segment. This is B segment. This is C segment. A, B, C segments are there throughout the axon. This is axon membrane. Okay. Throughout the membrane there is a now conduction and impulses going to take place. So, guys, if Na enter, it is already minus 70 MV, mean millivolts. Sodium ions are the pledge charge. Pledge charge. If the pledge charge enter into this segment, A segment, there is a changes in the electrical charge, mean minus negative. Negative charge going to decrease, mean the pledge charge is going to become minus 70 going to become minus 60 guys, minus 60 MV, minus 70 going to become minus 60 MV. In this condition, what happened? Mean the electrical charges are going to changes, mean minus 70 become minus 60. So, there is a voltage is change, voltage is change. We have to see in this graphical representation. Okay, we have to look into this graph wise. Okay, guys, this is graph. This is graph. Okay, this is minus 70. Minus 70, give you a side. This is 0, this is minus 70. Minus 70 is here guys, minus 70 what we can call resting potential. Resting potential is minus 70. Resting potential. Resting potential is minus 70. If any electrical volt change, if the sodium mechanical gated channel open, sodium ion trying to enter inside. The sodium ion is the pledge charge. If the pledge charge enter, the minus going to become reduce, that leads to the minus 60. Okay. In that condition, minus 60 going to like give the signals to the voltage gated channel. Mean if the voltage is changed, voltage gated channel open. If the voltage is changed, voltage gated channel is open. Mean minus 60 will give the signal into the voltage gated channel. This is the voltage gated channel for sodium potassium. If the voltage gated channel open, there are huge sodium enter into the A segment. Again there are number of sodiums are enter. Sodium enter. Number of sodium enter into the A segment. So this is to this minus 70 to minus 60 voltage gated channel, they can try reason for the open the voltage gated channel, mean they can activate the voltage gated channel. In that condition, more sodium ions are influx, mean they are enter into A segment, more like sodiums. So they can encourage this degree of changes which is minus 70 to minus 60. Whenever the changes of the voltage, they are activate the voltage gated channel. In that way, this is the sodium, huge sodium enter into the cell neuron. That is the condition. This is minus 60 going to become minus 40, minus 20. Finally, it is reached minus 20. Finally, it is reached plus 30, plus 30 up to plus 30. Guys, this is the first reason for this is minus 70 to minus 60 is the first reason for the opening of voltage gated channel. That is the voltage we can call threshold potential. Mean minus 60 is minus 70 become minus 60, minus 60. Minus 60 we can call threshold potential, threshold potential. The threshold potential is minus 60, is potential. Mean first initially this is guys first minus 70 to minus 60 it is threshold potential. Minus 60 it is become minus 40, minus 20. Finally, they become plus 10, plus 20, plus 30. They can reach up to the plus 30 guys. Mean here there is a huge amount of sodium enter, huge amount of positivity enter, mean the negativity decrease up to the plus 30. Okay. This is the condition plus 30 what we can call action potential, action potential, action potential, action potential. This is the cell excitement what we can call depolarization, depolarization. This is we can call depolarization. Depolarization. First initially resting potential which is always in the rest mode of the cytoplasm which is maintained minus 70 MV, which is we can call resting potential. If any mechanical gated channel open, there is a sodium enter, initially sodium enter, that leads to the there is a potential difference is going to take place minus 70 to minus 60. if voltage it is changes, any changes in the voltage gated channel open, that is initial changes we can call threshold potential, threshold potential, threshold potential, threshold potential. That is threshold potential will reason for the voltage gated channel open. If the voltage gated channel open, more sodium ion going to influx, going to enter into the cell. In that condition, more positivity build up within the cell, there is more positivity going to make up. In that condition, this minus 60 to become up to the plus 30. This is the condition we can call action potential. Action potential nothing but one wave complete, mean one impulse is complete for the action potential. Okay? The signal is started up to the plus 30. Guys, here a condition, sodium ions are enter quickly, or enters quickly through the channel. Potassium ions are they trying to exit out from this segment very slowly. Okay? Potassium ions are slowly exit out. Sodium ions are, if any channel open, sodium ions are very quickly trying to enter in. Potassium ions are very slowly exit out from this segment. What happened? Here there is a potential is different, going to change. In that condition, voltage gated channel open. In that condition, immediately the sodium ions trying to enter in. That is why the voltage is changes up to the plus 30. Electrical negativity going to become positivity. In that condition, there is a potassium gated channel is open. The potassium is trying to exit out once attain the action potential. Potassium is trying to exit out from the A segment. Potassium is trying to get out in the A segment slowly after action potential. And where here in this A segment, sodium ions are trying to move from the B segments. Sodium ions trying to move into the B segment. Sodium ions are trying to enter into the B segment. B segment also same as resting potential minus 70 MV. Minus 70 MV. C segment also minus 70 MV. If sodium ion come into the B segment, this minus 70 MV going to be changed into the minus 60 initially. Minus 70 going to change into the minus 60. This is threshold potential again. Threshold potential again. If sodium ion from A segment to they enter into the B segment, plus charge is enter into the B segment, and plus charge which is potassium is going to out. In this condition, there is a plus charge is going to move out from this segment which is in the form of potassium, and in this segment as well the sodium ions are trying to enter this segment. Two ways there is a positivity removing from the A segment, two ways. One is in the form of potassium they exit out, second one is in the form of sodium they enter into the B segment. Huge negativity is going to take place up to minus 90. This plus 30, the negativity going to take place up to minus 90. Up to minus 90 in the A segment. Up to minus 90 in the A segment. Here this depolarization, they has to come down up to the minus 90. This is the minus 90. Once the potassium ions move out, trying to move out, and sodium ions, they trying to enter B segment. In that condition, the positivity come this side and positivity come outside. In that condition, there is a huge plus 30 to minus 90 is going to take place, 90 millivolts. This is we can call repolarization. If again this B segment going to become depolarization up to plus 30. Again, how? If voltage is changed, threshold potential is to become voltage gated channel open again. If the voltage gated channel open, sodium enter inside, huge. If the sodium huge enter into inside, it is to become minus 60 to plus 30. In this condition, this is we can call action potential, again B segment. So, this condition, yeah, potassium is trying to move out, potassium through the potassium gated channel, potassium trying to move out, and sodium trying to enter into the C segment. If sodium enter, minus 70 to become minus 60 plus charge, if the plus charge enter. This is we can call again threshold potential. So guys, again it is to become repolarization in the B segment also. Again there is a, if the sodium come this side and potassium come out, that leads to the minus charge is going to take place up to minus 90. Plus 30 is going to become minus 90 here again. This is repolarization. Depolarization is continue in the, this is the question guys, NEET. Depolarization is continue in the wave pattern in the membrane layer. Below the membrane layer, depolarization is wave pattern and repolarization is continue along with depolarization. Repolarization is continuous along with depolarization up to the end of the axon. It is going to take place not middle of the cell, the below of the cell membrane. This is also NEET question guys. Okay? If the B segment depolarization, A segment which is followed, that is repolarization. First depolarization, then repolarization. It is wave pattern they can move. Now impulse is the wave pattern, okay, in the below the cell membrane only, Depolarization is continue the wave pattern up to the axon end, repolarization is followed. This is what about exam. Guys, you have to look into this graph. First minus 70 become minus 60 is the first step. This is first step to change the voltage. That is why it is a threshold. Threshold mean to encourage, okay, in the voltage changes. That is encouragement to open the leakage channel of sodium and potassium. That is potassium is trying to exit out, sodium is trying to like come into the next segment. In that way, huge positivity move from the A segment to B segment. A segment going to create a minus 90 up to the negative charge, more negative charge, that is repolarization. The more positive charge which is up to plus 30, which is action potential, we can call depolarization. Depolarization, which is up to the action potential. Action potential is what? One now impulse we can call action potential. One now impulse called action potential. Action potential. So what happened after the C segment is the axon end. Assume this is axon end, this is C segment. So in the axon end, yeah, I hope this graph is clear. Okay? Yes. If any, this is axon guys, this is cell body, this is axon. The signals are passed throughout the axon. So they can give the signal into the next neuron cell. Mean there is a gap. There is a gap between the neuron and the neuron. This is axon end guys. This is axon end. Axon end. And next neuron started. Next neuron dendrite started. Next neuron, this is next neuron. Between the neuron, there is a gap we can call synapse. This gap we can call synapse or synapse. So in the C segment axon end, there is a channel which is calcium gated channel, mean ligand gated channel are there. In the C segment, there is a ligand gated channel, mean chemical gated channel, which is calcium channel is there. Okay, minus 70, if minus 70 become minus 60, it is changes of the voltage. Okay, that leads to the calcium channel open. Calcium channel is open. There is a chemical vessel is there. Chemical vesicle is there. This is vesicle. Inside the vesicle there is a chemical we can call acetylcholine. Neurotransmitter ACH chemical is there. ACH chemical is there. We can call neurotransmitter. This chemical neurotransmitter. Neurotransmitter is acetylcholine. Okay, the calcium ion push this vesicle, the acetylcholine neurotransmitter, they can come out into the synapse place. Synapse place, they can come out the chemical. Okay, if the chemical is come out, they can activate this part guys. Next neuron, they can activate the channel chemically, ligand gated channel open here. If the ligand gated channel open, there are again sodium ions are enter here, pledge charge. If the pledge charge enter, there is already minus 70 MV in the next neuron, right? That is going to become change into the minus 60. Minus 60. So throughout the one neuron is throughout the impulse going to take place, and that can carry forward into the next neuron through this synapse. This place we can call synaptic cleft. Synaptic cleft. What is synaptic cleft? Mean the end of the axon. The end of the axon we can call presynaptic. presynaptic, presynapt. This is the next axon, the axon dendrites we can call postsynapse. Presynapse and postsynapse, the between place we can call synaptic cleft. They can cross the signals, impulses through the synaptic cleft into the next neuron. If the ligand gated channel open, the chemical which is released to open this channel. This channel, if the open, there is a sodium ion are directly enter into the inside of the next neuron through the ligand gated channel open. Guys, remember, first mechanical gated channel open first. After that, if the voltage change, voltage gated channel open second. Third, if the voltage huge enter, the sodium ions are next coming to move next segment and potassium ions exit out with the leakage gated channel. Third, finally, if the chemical release into the synaptic cleft, they can reach the this is the receivers, guys. There is a receivers, they can attach the chemical into the next body of the dendrites of this next neuron. That is chemical is reason for the again open ligand gated channel. Through this channel, the sodium ion enter into the next segment of this neuron. Next neuron which is open ligand gated channel. First initially mechanical gated channel, end of the axon, that is ligand gated channel open here in the next neuron. Okay. Yeah, this is calcium ions, calcium channel ions, they can open end of the axon, that is calcium ion push this vesicles which is filled by acetylcholine, that is neurotransmitter. Acetylcholine is the neurotransmitter, that is the chemical which is reason for the next neuron open that channel, ligand gated channel. In that condition, sodium is enter into that next neuron, that leads to the changes in the voltage which is resting of the -70 going to activate to change the voltage, that leads to the voltage gated channel open, that is threshold potential. Guys, in this pattern, the wavelength pattern, the now impulse and conduction is going to change from going to transfer from one neuron to next neuron. This is about the conduction of now impulses along with axon. Guys, I hope you did understand well. Okay? Yeah. If any challenges, please let me know with the comment, and if it is good, please hit the like button. This is NEET World for you to give the best teaching. Thank you so much.